Before CRISPR, genome editing was painstaking and baroque, dominated by zinc finger nucleuses and talons developed in the 1990s and 2000s. Those tools worked. Groups like Sangamo and academic labs showed targeted cuts in mammalian genomes, but bespoke protein engineering, long timelines, and high costs kept the techniques confined to specialist labs. The breakthrough began in 2007 when Rodolphe Barangou and colleagues showed in Science that Streptococcus thermophilus gains resistance by integrating bacteriophage sequences, revealing that CRISPR arrays function as an adaptive immune system. Emmanuel Charpentier, then at Umeå University in Sweden, dissected the small RNAs and tracer RNA that guide Cas9, while Jennifer Doudna, a structural biochemist at the University of California, Berkeley, and an HHMI investigator, worked out the RNA protein architecture. Together, their teams showed that a single Cas9 protein could be programmed with a short guide RNA to cut DNA at specific sequences. That simple idea, a programmable RNA-guided nuclease, promised to replace artisanal protein design with a universal, easy-to-use scalpel for genomes. With the technical background, the people involved, and the stakes for medicine, agriculture, and basic biology clear, the stage was set for the experiments that followed across Berkeley and Boston, experiments that quickly turned possibility into practice. On June 28, 2012, Martin Ginnick and colleagues published a short but seismic paper in Science, Ginnick et al., Science 2012, that turned the whispered promise of bacterial immunity into a practical biochemical tool. They showed in vitro that Streptococcus pyogenes Cas9, guided by short CRISPR RNAs, makes blunt double-strand breaks at DNA sites complementary to a 20-nucleotide guide and immediately adjacent to the required PAM sequence NGG. Crucially, they demonstrated that the natural dual RNA guides, the trRNA and tracer RNA, could be fused into a single simplified guide RNA that retained full cutting activity. From then on, a user needed only to change a short 20-ant sequence to redirect Cas9. The experiments were elegant and minimal. Recombinant Cas9 plus a synthetic guide produced sequence-specific cleavage in test tubes and gel assays, cleavage maps, and the PAM dependence made the mechanism clear. The modularity was obvious and revolutionary. Specificity was encoded in a short RNA, while catalysis lived in a single protein with HNH and RUVC nuclease domains, rather than in bespoke protein engineering. Within weeks, the community grasped the implications. Labs could design guides online, order oligos, and test targets in days instead of months. Subscribe. Apparently, it speeds things up. Conversations from Berkeley to Umeå to Boston and beyond shifted from cautious interest to rapid planning to adapt the system to eukaryotic cells and therapeutics. The contrast with zinc finger nucleuses and talons, which required artisanal protein-by-protein customization, became a rhetorical cudgel. CRISPR-Cas9 was simple, programmable, and scalable. That clarity turned curiosity into a sprint, and by early 2013, groups including Feng Zhang's team at the Broad Institute and labs at Harvard were pushing the RNA-guided scalpel into human cells and whole organisms. On January 3, 2013, Feng Zhang and his team at the Broad Institute in Cambridge, Massachusetts, published a pivotal paper in Science that turned Ginnick et al.'s in vitro scalpel into a working tool inside human cells. Zhang's team, human codon-optimized Streptococcus pyogenes Cas9, added nuclear localization signals, expressed single-guide RNAs from mammalian promoters, and showed efficient, sequence-specific cutting and gene disruption in cultured human cell lines such as HEK293. They also demonstrated multiplexing with lentiviral guide libraries and laid out practical workflows for designing guides, delivering reagents, and screening edits, turning a biochemical curiosity into a reproducible toolkit for eukaryotic genomes. The work was methodical and pragmatic. Plasmid maps, promoter choices, codon tweaks, delivery strategies, and percent editing metrics made the technique immediately adoptable by molecular biology labs worldwide. Within weeks, labs in Boston, Berkeley, Cambridge, and beyond were swapping protocols, reagents, and guide sequences. Within months, biotech startups and venture investors marshaled resources around CRISPR's commercial promise. That gilded scramble exposed a raw fault line. Who owned the right to use and profit from CRISPR in human cells? The Broad Institute filed patent claims focused on eukaryotic applications and moved quickly to commercialize. The University of California, representing Jennifer Doudna and Rna Charpentier's early work, asserted broader priority based on the 2012 GeneX paper. Competing patent filings, grant races, and startup deals cascaded into legal contests and proceedings at the U.S. Patent and Trademark Office, culminating in high-stakes hearings before the Patent Trial and Appeal Board in 2016 and 2017. Technical triumph and commercial jockeying advanced in lockstep, sharpening incentives and tensions and setting the legal and ethical battleground that Act 3 will confront. Those technical and legal breakthroughs set off an era of explosive activity. Between 2013 and 2018, labs worldwide published thousands of CRISPR papers, venture capital poured into scores of startups, and translational programs moved from mouse models to human trials. By 2016, early clinical experiments in China had injected CRISPR-edited T cells into cancer patients. Between 2017 and 2019, companies such as CRISPR Therapeutics, Editas Medicine, and Intellia Therapeutics launched clinical pipelines for blood disorders and rare diseases, and ex vivo trials for sickle cell disease and beta thalassemia began to move toward patient cures. The gilded promise of rapid therapeutics soon ran into hard practical limits. Off-target cleavage, mosaicism in embryos, inefficient homology, directed repair, delivery barriers to specific tissues, and immune reactions to Streptococcus pyogenes, Cas9. Those real safety gaps drove urgent ethical debate. A landmark summit in Washington, D.C., the International Summit on Human Gene Editing, December 1st through 3rd, 2015, urged caution and public engagement, and a 2017 U.S. National Academies report called for stronger governance and tightly circumscribed clinical use. The field was rocked on November 25, 2018, when He Jiankui announced in Hong Kong the birth of purportedly gene-edited babies. That announcement provoked worldwide condemnation, prompted a Chinese investigation, and led to a December 2019 criminal sentence that shocked colleagues and regulators. After 2018 calls for moratoria, the World Health Organization convened an expert advisory committee in 2019, and many nations tightened oversight. At the same time, patent battles and commercialization races continued. The result was mounting pressure. Scientists faced reputational peril, regulators scrambled to set rules, investors weighed risk, and the public demanded limits. Technical peril, ethical fury, legal strife, and regulatory urgency condensed into a single electric moment. Everything seemed poised to break one way or another. The high point came on October 7, 2020, when the Royal Swedish Academy of Sciences awarded the Nobel Prize in Chemistry to Rna Charpentier and Jennifer A. Doudna for the development of a method for genome editing. That announcement in Stockholm confirmed the significance of the 2012 GeneX et al. Science paper and the transformative promise of an RNA-guided scalpel that turned a bacterial immune system into a programmable tool. Laboratories celebrated, funders treated the prize as proof of translational potential, and the field drew wider public attention. But the award also exposed concrete disputes. Patent battles between the Broad Institute and the University of California, PTAB proceedings in 2016 to 2017, competing claims from Feng Zhang over priority and commercial rights, and unresolved questions about equitable access. He-Jianqui's 2018 rogue embryo edits and WHO advisory steps in 2019 had already made governance urgent, and the Nobel amplified calls for international oversight, transparency, and restraint. The prize acted as both coronation and summons, honoring a powerful method while forcing the community to decide who gets credit, who controls applications, and how to steward CRISPR as it moves toward human consequence. After the 2020 Nobel, CRISPR-Cas9 moved from a lab curiosity to a platform driving rapid development of therapies, diagnostics, and agricultural innovations. Companies like CRISPR Therapeutics, Editas Medicine, and Intellia Therapeutics pushed ex vivo therapies for sickle cell disease and beta thalassemia toward clinical success and accelerated in vivo trials. Follow-on inventions: base editing, Komor et al., Nature 2016, David R. Liu Lab, and prime editing, Anzalone et al., Nature 2019, David R. Liu Lab, opened subtler, less disruptive ways to rewrite single DNA bases without double-strand breaks. Diagnostics and gene drives expanded, while WHO and national regulators convened ethics and governance forums. WHO Advisory Committee on Human Genome Editing, 2019. But persistent technical hurdles, efficient delivery, off-target edits, and immune responses, and societal challenges around access, equity, and oversight will determine whether the 2012 insight becomes an unambiguous boon or a contested legacy.